Hi, let's talk about the nervous system with the cranial nerves for head and neck anatomy. So I'm going to, well, I guess we'll start out with credit for some of the slides that I'm going to be using. And this is what I want to do first for this lecture. It'll be a basic review of some of the nervous system components that you saw in A&P. So I'll go ahead and review that, and that will be it for this lecture. So let's go ahead and get started. So before I do this, let me back up and just do a sort of a broad overview of the nervous system. So it's useful, I think, to view this nervous system as, or these cranial nerves perhaps, as a stimulus response system. That's basically a lot of the nervous system, is that different stimuli come into the brain. Sight, sound, taste, smell, and then the brain decides what to do with it. Is it something to be excited about? Well, then we need to deal with it. Or is it something that can be ignored? Like the noise from the fan in another room. You can ignore that, it's not critical. If all of a sudden there is a dog barking and you don't have a dog, well, that's something that may require a response. So if you poke the tongue with a probe, or you accidentally pinch it or jab it with a probe, well, there's a pain stimulus and the patient's going to respond. You know, their muscles will contract, they'll pull backward, they'll flinch, etc. So all of these cranial nerves are involved with these stimulus response reactions. So try to keep that in mind, and they form a loop. So here's some sort of stimulus, stimuli plural, and that information is sent. Now this is often on your tongue or your fingertips or your eyeballs or your nose or wherever this sensory information is coming from. So this is the sensory info, and it's traveling through sensory nerves, sensory nerve fibers. And then it comes to some sort of integration center, which is our brain. The brain decides what to do about it. Ignore it, or get out of the way, perhaps. So let's just call this brain. That's good enough. And then the brain decides what to do, and then the brain sends out information through motor nerves. And that goes to some sort of effector. And effector is sort of a general term for what's causing the effect. And typically, let's say, for example, the effectors are muscles. And they move in a coordinated fashion as controlled by the brain. So you have this loop. Now you have the stimulus, and then there's this loop, and then you have this, which is our response. Okay? So let's go ahead and get started on this next slide. But keep this picture in mind, because basically the next slide is putting the details into this basic picture, or a few details. So here's I guess what I like to call an organizational flow chart of the nervous system. So, just to add a little bit here, CNS is for central nervous system. This is going to be our brain and spinal cord. That's the CNS. So the medulla oblongata and the rest of it is all over here. So that is our CNS, brain and spinal cord. If it's not part of the cord, it's not part of the CNS. So let's go and let's start with sensory, because it's driven by our responses should be driven by sensory information. So let's just put in here our 5 basic senses that most people learned back in grade school somewhere. Sight, hearing, taste, touch, smell, etc. And let's say perhaps let's put in here, that's good enough. Let's just leave it alone. And we'll come back to this idea of the number of neurons in a pathway. But all sensory neurons, all sensory pathways are a single neuron until you get into the CNS. And then the number of neurons, of course, explodes. So let's do this. Let's send sensory information through the PNS into the brain. It takes one neuron from the taste buds of your tongue into the brainstem, spinal cord, or somewhere in the CNS. There's one sensory neuron. When you step on a tack with a bare foot or those goat head thorns, there's one sensory neuron that runs from your foot up to your spinal cord. So all sensory neurons are a single neuron in that pathway. And then coming out, the brain decides what to do again. And let's take an easy one. Let's just come out. This is going to be motor. Comes out, and then let's just have it loop down to this box. So this is our motor output. So we could define these sensory neurons as afferent and the motor as efferent. So A comes before E. So sensory information has to go in first, and then efferent is motor coming back out. And all of this, again, is part of the PNS, which includes spinal nerves and cranial nerves. So some terminology. When we're talking about somatic motor, these are voluntary skeletal muscles. Abbreviate skeletal muscles. MM for muscles. And those are stimulated with each action potential that arrives at the muscle. Stimulated to contract. Abbreviate with each AP, or action potential. Okay, so we did a lot of that in ANP1. So that's one motor pathway. And to somatic motor, to somatic skeletal muscles that are voluntary, it's always one neuron. after you leave the CNS. So the muscle or the nerve that carries impulses to the muscles in your foot, it's a single neuron that leaves the spinal cord and travels all the way down to your foot. Okay. Or maybe it's a single neuron that's going to your temporalis muscle or your masseter or any of your muscles of facial expression, because those are all voluntary. All right, keep on with this review. Other motor outputs are not voluntary somatic. They go to, they are autonomic. So maybe I'll just draw another, maybe I'll do blue here. Cross over the green and go to the autonomic. And these are all 2 neuron pathways, all autonomic. And autonomic is going to include both the parasympathetic and the sympathetic that we'll get to. So the second neuron goes to here or goes to there. So let's be pretty clear on what we're talking about with the autonomic nervous system. If we're talking autonomic, one thing that you should appreciate pretty quickly is that if we're talking autonomic, we're talking motor. So we have somatic motor and we have autonomic motor. So autonomic is just motor output, and it's going to, these are the effectors, if you remember from A and P, smooth muscle, cardiac muscle, the heart, and glands. Lacrimal gland, salivary gland, etc. Okay. So we will be, for your class, we could be talking about glands, salivary glands, lacrimal glands. For smooth muscle, what we're going to be most concerned about is vascular smooth muscle in the blood vessels, because you are interested in controlling bleeding with certain dental procedures. Okay, so just to, again, kind of wrap this review up, remember that parasympathetic is rest and digest, and sympathetic is fight or flight. But in both cases, we're still talking about the same effectors. And those effectors, again, are smooth muscle, cardiac muscle, and glands. So these, again, are the effectors. They're what will cause the response, the effect. And again, this is these are rest and digest functions. And these under sympathetic are fight or flight functions. Survival. So, for example, both fibers go to the heart, cardiac muscle. Parasympathetic fibers would be lowering the heart rate. If the sympathetic is dominating, we would be raising the heart rate. if we have dual innervation to, and we do, to salivary glands. This is a target of the autonomic nervous system. You don't have voluntary control over them. If the parasympathetic pathway is dominating to the salivary glands, what would the response be? More saliva for digestion. If it's a sympathetic response, and the sympathetic pathways, sympathetic neurons to the salivary glands dominated because we're scared, we're in a stressful situation, a fight or flight situation, what's going to be the response on the salivary glands? It's typically the opposite of the parasympathetic. So you'd wind up with a dry mouth, less salivation. Okay, that's enough of this slide. Let's move on. And here we can talk a little bit about neurotransmitters and receptors. So again, keep in mind when we're talking about motor neurons or motor pathways, if we're talking about somatic motor, these are voluntary skeletal muscles. All of those that you just learned for muscles of facial expression, muscles of mastication, the hyoid muscles, supra and infra, they're all somatic. Again, it's a one neuron pathway, and the neurotransmitter that is released is acetylcholine, which we will abbreviate as ACH. This is review from last quarter, and it is released from the axon terminals. So these are axons, of course. And the receptor that's on the effector, so again, the effector here would be skeletal muscle cells, is a nicotinic receptor. So I sometimes abbreviate this as ACH for the neurotransmitter onto a nicotinic, and I'll just abbreviate NIC receptor. That's what occurs in this synapse between the motor neuron and the effector. That's the little gap that we looked at a couple quarters ago. Okay, so that should be review. Parasympathetic, let's do the same thing. We have two neurons now, one on the left, one on the right, and here is the ganglion. So if you remember, the definition of a ganglion is a collection of cell bodies, CB, cell body, and these of course are the axons. So here's the other cell body, CB, and the axon. So there are our two neurons of a parasympathetic pathway. And this is the parasympathetic ganglion. They're outside the CNS. They're somewhere in the head, neck region. And it's a collection of multiple cell bodies, which is our definition of a ganglion. So the neurotransmitter receptor combination is exactly the same as somatic motor. So the first neuron out of the CNS, because these cell bodies are in the CNS. So this is CNS, and this is PNS. The first neuron leaving is always a cholinergic neuron. It's always going to be releasing ACH from the axon terminal. So this also would be, I'll just abbreviate now, releasing ACH from the preganglionic neuron. preganglionic neuron. So that's always a cholinergic neuron releasing ACH, acetylcholine. And the receptor on the postganglionic cell is nicotinic. Again, it's always that same pattern. So this is the postganglionic, I'll abbreviate, ganglionic cell. So we have a pre and a post, one before the ganglion, one after the ganglion. All right. The effector now for the second neuron, keep in mind that we're dealing with parasympathetic right now. The second neuron also releases ACH. But it is released onto a muscarinic receptor. Which is sometimes just abbreviated as MUS. Okay? Remember what the effectors are here. Smooth muscle, cardiac muscle, and glands. Okay? We will be dealing with parasympathetic pathways more than sympathetic pathways. Mainly because parasympathetic fibers run in cranial nerves. Sympathetic fibers are not found in cranial nerves. They travel by other means. So that's our second pathway for parasympathetic. Let's do sympathetic. And again, two neurons. Here is our ganglion, again, pre and post ganglionic cell. And it's the same pattern for the first neuron. It's ACH onto a nicotinic receptor. And keep in mind, the nicotinic receptor is on the postganglionic cell. So the neurotransmitter is going in that direction. Okay, so the first neuron is always the same. The postganglionic on the effector, again, we're talking about smooth muscle, cardiac muscle, and glands. The postganglionic neuron is releasing norepinephrine. I'm going to abbreviate that as NE. So not epinephrine, it's just norepinephrine only. From sympathetic neurons. And then we have a couple of different receptors that are possible. And you may remember that we had beta 1, beta 2. We're going to just put in beta 1 only, and alpha receptors. Okay. So let me do another slide here because I'd really kind of like to stress this a little bit more. So let's go to another page and we'll just try to summarize this. So these are adrenergic receptors. They bind norepinephrine or adrenaline, noradrenaline. Adrenergic receptors. So we are dealing with the autonomic nervous system, sympathetic So the adrenergic receptors that we are dealing with are beta 1, and these were found primarily on the heart. We're going to keep this as simple as we can. That we did last quarter. Beta 2 was on smooth muscle in the lungs, specifically on bronchiolar smooth muscle. And this is important because when the beta 2 receptors on bronchiolar smooth muscle are stimulated, it causes the smooth muscle to relax and the airways dilate so you can run faster. And that leads to bronchodilation. Okay, so opening of the airways. So both, well, let's do the last receptor type. And there are several subtypes, but we're going to again keep it simple. And we're just going to do, we're just not going to worry about subtypes. We'll just call them alpha receptors. And these are the most common. And what I would like you to remember is that it's alpha receptors on vascular smooth muscle. Okay, all of these will bind norepinephrine and epinephrine. So all will bind NE, norepinephrine, and E for epinephrine. Norepinephrine is released from sympathetic neurons. The postganglionic symp neurons, and let me do a different, sorry, let me scratch that. Let me do it this way. Let's say that sympathetic neurons release norepinephrine. And these are the postganglionic. Release norepinephrine only. The adrenal medulla releases both. The adrenal glands. Both NE and E. And all of those, or both of those, will stimulate all of those receptors. So I need to wrap this up, so just bear with me. The thing that I would like you to remember is that when you administer anesthetic agents, anesthesia, local anesthesia, you are controlling pain and bleeding. So it's usually a mixture of something like lidocaine or novocaine to control the pain, and there's also oftentimes epinephrine in that solution. So next time you're drawing up a syringe for somebody that's for a procedure, take a look at the vial, and it should say lidocaine or novocaine or some other analgesic with a concentration, but it also should indicate, or they often have epinephrine involved also. So why put epinephrine into the injection for pain? Because it binds to the alpha receptors, binds alpha receptors on the vascular smooth muscle in the area, the blood vessels, and causes vasoconstriction. So we have increased vasoconstriction, less blood, less bleeding. I don't want to go into it now because I feel like I've already done more than I really wanted to. But if you have a patient that has allergies, severe allergies, and they're carrying an EpiPen, if they have something like an anaphylactic reaction and they're carrying the EpiPen for that, you administer epinephrine, and that binds to the alpha receptors, squeezing the blood vessels and raising their blood pressure. It also binds to the beta-2 receptors on the lungs and causes bronchodilation and opening of the airways, which is what is going to kill somebody with an anaphylactic reaction if it gets out of hand, is bronchoconstriction and crashing airway or crashing blood pressure. Okay, let's move on. Okay, so just review, and I won't spend as much time on this, but this might help with your anesthesia section that you all are doing also in fall quarters. So just to remember, the sodium-potassium pump, it's on the membrane, and here is the pump in this picture, this green and blue structure, and remember that it's pumping 3 sodium outside the cell into the extracellular space, simultaneously bringing in 2 potassium into the cytoplasm. And this pump requires ATP hydrolysis. So there's ATP being hydrolyzed to ADP and P. It takes energy. And that sets up these gradients with sodium, high concentration of sodium on the outside, and so up arrow, and a relatively low concentration or a small sodium signal on the inside. They just put different size fonts. It's the reverse with potassium. Most of the potassium is inside cells. There's a little bit of it on the outside. That sets up this chemical gradient. The other piece of this sodium-potassium pump story that sometimes doesn't get discussed much is the potassium that leaks out. So we have more potassium inside and less potassium outside. The potassium flows out through these leakage channels. So if you can remember, let's just say remember, as as potassium flows out of cell, cell becomes more negative. Okay, so here's the result of the sodium-potassium pump running in the picture above, and here's sort of the end result. Outside of the cell, as you can see, is positive, and the inside of the cell is negative. Notice, though, that this difference is only at the membrane. So this is our membrane potential. Our electrical potential. It's only at the membrane. If you look deep inside the cell, it's neutral. The charges are canceling each other out. And the same outside. Okay. So if you measure the difference in millivolts, it looks something like this. The inside of the cell is slightly negative at about minus 70 millivolts. And up here maybe it would be 0. And if you kept going, maybe up here it would be, I don't know, plus 20 millivolts. So if it's at rest, it just is sitting here. So that's our resting, not doing anything, membrane potential. Typically around minus 70 millivolts. And this is our electrochemical gradient. Here's the electro part, and the chemical part is the sodium and the potassium. Okay. The action potential, just to kind of buzz through this a little bit. Here's our RMP again. We're sitting at minus 70. And stimulus comes along. If it's a weak stimulus, it may fail to initiate an action potential, and we sometimes call those graded potentials. Nothing happens. We didn't hit threshold. Once we hit threshold here, then we have a rapid depolarization as sodium ions move into the cell. And that creates the AP, a spike. So we went from minus 70 past 0. We're up here somewhere around plus 40. I don't care about that number, but that's the idea. And then the cell has to repolarize quickly, so potassium ions leave. And what did we say potassium ions do to the membrane potential? They make it more negative. So as the potassium ions are leaving, the cell is becoming more and more negative. We're going down the vertical axis, and we're going quite negative. There's this little bit of undershoot, and then we come back up to rest. And we're back up to our resting membrane potential. Okay? That's pretty much review. Note that this happens pretty quick. Start to finish about 2 milliseconds, 2/1000ths of a second. So the next slide is just basically the same thing, but it's just a little more detail on what's going on with the ion channels. So right here is where the stimulus arrives. So there's our stimulus. And sodium channels open. And more sodium channels open during the green. So we have a very rapid depolarization as lots of sodium channels open, sort of by feedback, positive feedback. They then close near the top, and then potassium channels open, and during the blue. So all the while during the blue, the potassium is flowing out and repolarizing the cell. Okay? That should be reviewed. The last thing that I thought we might want to do is just be to remind you of what is going on an axon. So if this is our axon, remember that there are gated ion channels along the length of it. And there are little trap doors. Some of these are sodium channels and some of those are potassium channels, but they have gates on them. And they're not literally in pairs, but that's just how I'm drawing them. And they're all scattered along, you know, both sides, of course. Oops, I can't draw upside down very well, I guess. So, you know, little trap doors are the gates. So these are gated sodium and potassium channels. That's what was opening and closing. So we just looked at gated sodium and potassium channels. You may also remember that on most of these neurons that we're looking at, sensory and somatic motor, they're very fast. To get a fast stimulus response going, they are myelinated. And you have these myelinating cells that wrap around bits of the axon, little segments, and there are multiple layers. And this forms our myelin sheath. There's just one segment, and they're scattered all along the axon. So this is our myelin sheath. And then at the nodes, or before and after each of these cells, again, you have gated sodium and potassium channels. And that speeds up the conduction of action potentials. And that's all I wanted to remind you of. Speeds AP transmission. They travel faster, significantly faster. Okay? So that's enough of the review. I would pay attention to the neurotransmitters and the receptors. Pictures like that will help. We're not going to do a lot with it. We have other things to focus on, but I think it's good to remember that. Okay, so there'll be some questions on that. Please ask if you have questions. I'm going to give you homework to practice. Okay, I'll see you soon. Bye.